undergraduate University in Montreal. He did his master's in Oxford, and then his PhD in Eric Alves' lab at MIT, and then his postdoc with Pardeep Savani studying Lassa virus. And then he returned home to Montreal to start his lab, where he's now associate professor. And I wanted to comment a little bit about Jesse's very prolific body of work. If you haven't read any of his reviews, they're just amazing. He's really gone into depth into the ecological and evolutionary dynamics, microbes, and natural populations. And I've heard him speak in the past, and he gives a really amazing talk. So it's a great pleasure to have you here today.
Thanks, Anita. Thanks, Gary. Do we turn on my switch? Is that on? Yeah, good. Okay, excellent. Thanks so much for the invitation. It's really a pleasure to be here, particularly to escape a bit of just the Northeast winter. Also have a great morning, and I'm sure a great afternoon to talk to many of you here. So the story I'm going to tell you today is a question. Does diversity beget diversity in microbiomes? So first, it's really going to be one story today, just a little bit more background about what my research focuses on. And I study evolution and ecology of microbes. So when I talk about evolution, this is broadly changes in allele frequencies, mutations, changes that are happening within species. Ecology, these are changes or interactions between species, changes in relative species abundances and so on. So the ecology and evolution department, so hopefully this makes some sense to you, but this is like a really simplified version of my view of things. And then of course in this intermediate is, well, what is a species? And this is this sticky question that I think a lot of debates in biology kind of spiral to and end up being a species debate over, are we studying ecology or are we studying evolution? And I think in microbes, where evolution can be rapid, this is a particular sort of sticky area that I won't delve into here in particular, but is sort of one area of my research. And then more tangibly, what kind of things? So I study microcystis. It's a cyanobacteria that causes blooms in fresh waters. I study pathogens like maybe cholera. I did a bit of work during the postdoc on Lassa virus. Ongoing collaborations on tuberculosis, Haemophilus influenza, and other pathogenic bacteria. In terms of ecology, I'm interested in how some of these focal species of interest interact in their community context. So microcystis, as part of this cyanobacterial bloom community, which is actually quite diverse. Vibrio cholera is a diarrheal pathogen, so how it interacts with members of the microbiome, and so on. Okay, so speciation, origins of species, E2 after Darwin, 1859, so this sort of tree figure, branching process, right, where we get variety within species, kind of shows this kind of within species diversity, and then you get these branching events that lead to divergences that are sort of good, well-separated species. So when we think of diversification, this kind of evolutionary branching in a tree. So what controls the rate of branching, the rate of speciation or diversification more broadly if we want to consider sort of more ancient divergences? And there's quite a lot of literature on this, so people think of abiotic factors, things like temperature, pH, latitudinal gradients of biodiversity, right? So these sorts of things. And also biotic factors, so things like competition, mutualism, predation, species interactions that might affect the rate of speciation diversification. So there's two broadly competing models for how biotic factors would be expected to affect diversity. So if we plot on, and this will be sort of, I'll show a lot of plots that have these similar kind of axes, so I'll take a minute and kind of walk through what I'm talking about here. So on the x-axis here, we're going to be the initial diversity in the community. And on the y-axis, we have the diversification of a focal species. So basically, will that species speciate into two? Will it accumulate mutations or levels or polymorphism in the species? So how will the community context affect diversification of the focal lineage of interest? There's sort of two competing models. So one is what I'll call an ecological controls model, which is essentially saying that as we have a lot of diversity in the community already, there aren't any niches left, right? So there's no room for new species because all the spots are taken. So this predicts a negative relationship. And this was actually, this sort of niche filling model was included in an early edition of Darwin's Origin of Species. Where he used a metaphor, which I think is not very good, which is why it was removed from later versions, which was driving wedges into a log. And basically the log is only so big, and there's only so many niches, and if you jam in one wedge, another one has to pop out. So there's just sort of a finite number of niches, and maybe this is Darwin as being, you know, a sort of upper-class gentleman who had never driven wedges into a log, and in any case, this metaphor didn't really resonate, but I thought, you know, just as a historical tidbit, if for some reason, if that helps you sort of visualize this ecological controls model. Okay, so the alternative model is what I'll call diversity begets diversity. This is a sort of positive feedback, where actually the more species you have in a community, they're actually constructing new niches through predator-prey interactions, through cross-feedings, through a variety of different mechanisms that will then further beget diversity. Okay, so these are sort of our two competing models. And this terminology of diversity begets diversity, ecological controls, you could call this niche filling or something like that. I'm using the terminology from Dahlschluter and Handel from a couple of years. So I'm following this terminology. Okay, so evidence for these competing models. There's been a nice body of literature from experimental evolution, so looking at microbes in petri dishes, or flasks, which is cool because you can really control the background community and look at diversification of Pseudomonas, for example, in real time. So this is very nice, controlled experiments. And then there's been a reasonable body of literature on various different charismatic macrofauna that many of you may study. So what have these studies shown? Well, pretty sort of ambivalent and conflicting evidence. So these are studies in microbial microcosms, so niche occupation limits adaptive radiation, real-time microbial adaptation in the soil. So these are evidence for sort of a niche filling, the controls model. But then we also have other experiments showing the opposite, right? So high diversity stimulates diversification, showing that it's very deep. In more natural systems, we also have sort of conflicting studies or evidence for both models working in different systems, maybe on different time scales, including papers by some people maybe in this room or in this department, so on fishes and in Himalayan salt worms showing niche filling. But then other papers on reptiles and insects, some plants, showing evidence for a DVD model. Okay, so that's great. If a little bit ambivalent. So I posed the question, what about bacteria? From my perspective as a microbial ecologist and evolutionary biologist, well, microbes are the majority of genetic diversity on Earth, so we don't just really care about what model do they tend to follow, because that actually is sort of predominant genetic diversity on Earth. So what about bacteria in the wild, not in the test tube? So to do this, I started having conversations with a PhD student in my lab, Naima Amani, and they were both a collaborator at the University of Exeter, and we sort of hatched this project of how do we address this question. And we looked sort of through the literature and kept playing with different data sets, and finally we settled on this, which is the Earth Microbiome Project. It's a fantastic resource, a communal catalog of Earth's microbial diversity. It's a fantastic resource that we decided to use as our main test data set. So what the study did is very standardized sampling and 16S ribosomal RNA gene amplicon sequencing. So across the world, taking samples of different environments and sequencing 16S RNA gene, which is a highly conserved marker gene that's universal to bacteria and archaea. So it's a nice taxonomic marker of diversity. It's all done in a very standardized way, so all these samples are comparable. In this data set, there's about 155,000 different bacterial taxa, and this is at the level of amplicon sequence variants. So these are just unique sequences. You can think of these as species. We don't really know. They're basically just unique sequences, which we can group at taxonomic levels of genus up to phylum. And we worked importantly on a rarefied data set, which basically means we downsample every sample to exactly 5,000 sequences. So the sampling effort in each environment is equivalent, which is important because when you're looking to detect a diversity begets diversity relationship, you don't want to just find a relationship because you've sampled more in the environment. So we control for that, and we use a total of 2,000 samples. And where do these samples come from? It's fairly nicely distributed across the world. And these include, and they have this nice EMP environmental ontology, something like that, where basically we divide the samples into free living or host associated, or EMP ontology, anyway, not important. So free living from saline or non-saline environments, host-associated animal plants, and then down to 17 different biomes, soil, sediment, plant surface, and so on. So we know what kind of environment these came from. It's a really broad sampling of biomes across Earth. Okay, so how are we going to distinguish our DBD versus CE? So first, I'm going to sort of consider an evolutionary diversification model, and then show you why that's maybe not really the right model to be considering. I think it's because most of the literature has been really this looking at speciation, diversification. We're going to start with this. Okay, so again, we're looking at the initial diversity in the community. So let's say the number of genera, excluding a focal lineage of interest, and then we're going to look at the diversity within that focal lineage. So if in one sample, let's say that we have each branch, each long branch here corresponds to a genus. In purple is our lineage of interest, which then gives rise to two species. In this case, it's ASVs, okay, within that genus. So we plot that as a point, right, as one non-focal genus, and then within our focal genus, we have two ASVs. And in another sample, this is showing the DBD pattern, we have three non-focal genera, and within our focal genus, this gives rise to four ASVs, so we plot that point, draw a line through it, it's positive slopes, that's the DBD pattern. The alternative is basically the opposite of that. That's going to be consistent with a niche filling or a EC model. Okay. And you can do this at the ASV to genus level, for example. Okay, so that's an evolutionary diversification model. Probably not realistic at the resolution of our 16S data because these genes only accumulate a mutation every million years or so, probably, in terms of fixation. So we're probably not looking at de novo mutations within a soil sample or within a human gut sample. So probably we're considering something that's more of a community assembly model. It hasn't really been considered as much in this sort of DBD literature. So this is kind of how it would work. Where we start with a sample that has one genus. And then members of our focal lineage of interest are going to migrate, are going to arrive into that sample. And depending on the initial diversity in that community, a different number of different ASVs within our focal genus will find a niche in that sample. So in the DBD model, the more standing community diversity there is, the more room for new diversity to come. And under the DC model, this is like a niche filling model where we have something that's more like a carrying capacity of just 5 ASVs, right? So if we start with 1, we can accommodate 4 more. If we start with 3, we can accommodate 2 more. Okay. So this is just to show that I'm going to be a little vague about whether this is diversification or community assembly. We don't really know, but essentially the patterns can accommodate both processes in this case. Okay, so back to our sort of diversification model. So we can do this at the level of ASCs for the genus. We can sort of back down the phylogenetic tree, do genus per family, family per order, class, class file. Okay, we can sort of back down these taxonomy models. So we can consider these different levels. And we're going to do this in the context of generalized linear mixed models. We're going to model the number of ASVs in a focal genus as a function of the number of non-focal genera in the community. And we can add some, as a fixed effect, we can add random effects, including the sample ID, so just sample-specific effects, the biome types of soil, animal, etc., the genus ID, right, so the taxonomy or lineage, and the laboratories, because different people from around the world submitted these samples, so there's sort of batch effects and so on. And these results are pretty new. So there's a preprint on this. It's actually been updated since this was really, we're very much in progress, but I'm going to give you the latest version. So to simplify a little bit, we did not find strong effects of sample ID in laboratory, which is nice. So there aren't too many of those nuisance effects, but even if we can control them. But there are some effects of a particular biome or environment and the lineage, which is not unexpected. And the predominant pattern that we see are the positive slopes that support it. So this is just an example. At the phylum level, so this is deep taxonomy within proteobacteria. And proteobacteria is the most prevalent phylum, so this is somewhat representative. And across, each of these plots shows a different biome. So animal, proximal gut, water, plant rhizosphere, and we tend to see, so red are significant slopes, and they tend to be positive. Blue are not significant, so we see, in this case, no significantly negative slopes. And if we sort of go across all biomes, really most of them are positive across lineages and biomes, particularly at the phylum level. And all the positive, as we go down to the finer tips of the tree, ASD genus, there's a few more negatives, but DBD's positive slopes does tend to be the predominant signal. There are some negative slopes, so in some combinations of a particular biome and a particular lineage, we see significant negative slopes. So for niche filling, so this is Clostridium in non-saline sediments, we see significant negative. So it's not that we never see negative slopes, but the positive slopes do seem to be the predominant pattern. Okay, so all right, what else can we explain this, right? So we see these patterns and we try to dissect them. Okay. So data structure, right? Is this just, so we rarefied, right? So we're not dealing with differences in sampling effort, but could we get these relationships artifactually? And this is something, honestly, we're still struggling with and trying to perfect. It's a bit difficult to rule out effects of data structure, but so I'll show you what we've done so far. Taxonomic bias is another one. So so far these results are based on taxonomy, right? So it's saying this is the ASB in the genus Clostridium, but in bacterial taxonomy, there's a lot of unknowns, so we're missing things, and then different genera, or different phyla can have different phylogenetic depths. So it's not necessarily apples with apples in terms of tree depth. So can we do it differently to remove problems with taxonomy? And finally, abiotic drivers, right? So we're kind of considering these biotic effects, but could this be driven by abiotic factors as well? So I could consider all of these. Okay, so data structure. So we consider two different null models where we're basically going to permute our data. And this is what our data table fundamentally looks like, right? So we have these rows, which are different samples, so human gut or soil or whatever. And then we have counts of these different ASVs, different microbial taxa across samples. These counts actually have abundance information, but actually, for our purposes, we can just consider the presence analysis. So consider null model A, where we shuffle ASVs within samples, and this preserves the richness within each sample. And model B, where we basically shuffle ASVs across samples, so within a column, and this preserves the prevalence of ASVs across samples. So both of these have been used in the ecological literature and are sort of plausible nulls. Which is better or more realistic? I don't know. It's sort of debatable. And they give us very different results. So model A always gives a strong positive slope. Model B is pretty flat. Okay, so essentially, according to model B, our results of generally staying positive slope are highly significant and unexpected. According to model A, they're completely uninteresting. So we're still looking for ways to improve this and sort of select which is a better null and construct as opposed to permuting, but to do more generative models. So we're working on this. So we can't exclude this model A. However, looking at other synthetic data sets, which I'm not going to talk about in detail, we find that model A lacks power to correctly reject the null hypothesis when DBD is there for real. So this test is probably underpowered. Can't reject this model, but it becomes a bit sort of where it's possible that just samples with different levels of diversity could produce a DBD pattern, but it's sort of impossible to decouple those two things, or so it seems. So this is an important caveat to keep in mind. Also, this is with our whole data set, so it's not really modeling what we did with our GLMs, where we're including sample effects and biome effects that do account for some differences in diversity between samples, so maybe some of this can be accounted for. Honestly, we're still working on getting this just right. Okay, so taxonomy, right? So as I said, bacterial taxonomy can be a mess. Okay, so let's not rely on taxonomy. We're using marker genes, so let's just use that gene sequence information. So we're going to use different nucleotide identity cutoffs to delimit sequence clusters. So instead of talking about genus or class, we're just going to say 97% identity or 99% identity, and just kind of walk down the tree that way. And doing this, we also generally get significant positive slopes. This is a couple of examples. So looking at 97% clusters per 95% cluster in the community, or 100% per 97. I've also broken this down by biome. This is everything cool. I'm not going to show you everything, but the salient factor here is the slope of a linear fit. So the linear fit is gray, right? So positive slopes, but then we can also do a quadratic or a cubic fit, which shows, I mean, you can just see this looking at the raw data, is that it kind of goes up, but then it looks like there's a bit of a plateau, right? So it seems that at higher community diversity, we do hit a plateau, which is expected. You don't really expect this positive feedback of DBD going on indefinitely. Eventually, you are going to run into presumably niches becoming important. So to delve into this a little more, we broke this up by biome, and this is more or less what we expect, where we plot. So each point here is one of our 17 different biomes. This is at the phylum level, so community diversity phylum level, and you can see this nice relationship where we have pretty high DBD slopes when there's less diversity, but in more diverse samples, there's less DBD. So again, this is consistent with reaching a plateau as we just saturate with diversity. And the color coding here are host-associated biomes in black, so plant or animal basically, or free living, so aerosols, water, or soil. So the free living communities tend to be more diverse and tend to show lower DBD or slopes getting down to almost zero, so almost no DBD. Plant rhizosphere is an outlier, and that's probably because it's kind of like soil. Hard to disentangle what's really associated with the plants versus the soil. Okay, so the third thing we looked at are ebiotic drivers. Okay, so could these patterns actually be explained by abiotic drivers of both diversification and community diversity? So we looked at a subset of our Earth microbiome data, 192 samples that had data for pH, temperature, latitude, and elevation, which we know from the microbial ecology literature are drivers of baby diversity. And we just added them to our model, so we had these ebiotic factors, and we can test their effect. Okay, so we can do this again, ASD per genus, all the way up to phylum level. So look at what's the effect of community diversity in black here, and then what are all the effects of all these ebiotic drivers and their interactions. And again, so in red, this is kind of heat map, so we see this strong positive effect of diversity, getting stronger, kind of deeper into the phylogenetic tree. And then we do see some effects, likely positive or negative, of different abiotic factors, but really, diversity seems to be the main driver of diversification, with some effect of abiotic drivers, but that they don't seem to be the main drivers. Broadly, in the EMP, at least in this subset of samples. So we thought, okay, but what about soil? And we thought about soil in particular, right? So soil is quite diverse, which we know. And has been pretty well studied, right? So we actually considered another data set of 237 soil samples with quite a lot of environmental metadata, more than just pH, temperature, but really quite a lot of characterization of the nutrients and composition of the soil from this publication from a couple of years ago. And to summarize those results, DBD slopes are still generally positive, but no abiotic drivers, things like pH and latitude, have effects that are of similar magnitude as community diversity. So when you do look at these more diverse communities like soil, it does seem like abiotic drivers tend to take over. So I think that's kind of what I just said. So more diverse soil microbiomes are kind of at this plateau of DBD. They're very diverse, and it seems that abiotic drivers start to play a greater role in shaping diversity. So the next question we asked is, does the DBD relationship differ between habitat specialists and generalists? So this was a little bit inspired by this paper that came out a couple of years ago, in which they infer that generalists, so basically species that are able to inhabit a variety of different environments, are inferred to have higher speciation rates. Generalists, okay, they have higher speciation rates. Would they also have higher DBD? Would that speciation rate be more or less dependent on the diversity? I think you can sort of make arguments either way, that maybe specialists have higher diversification rates because they have a longer history entrenched in a particular environment, it's unclear which direction it will go. So we asked if basically generalists or specialists, which ones have higher DBD. So first we want to define some broad environmental categories. So we have these 17 different biomes, but we're going to reduce them down to 3. So we can naturally cluster them into 3 using k-means. So you can see here there's PCA, there's a pretty natural clustering of these three different environmental clusters, which basically are non-saline, saline, and animal. Okay, so these are our broad environmental categories. And then the circles here are at the genus level, the taxa that have a particular preference that tends to be found in those types of environments. So we're going to define resident genera that have a strong preference for a particular habitat. And then in gray are these sort of generalists that don't really have a preference. Okay, so then what we're going to do is look at our DBD relationship. So how does the number of resident genera affect the number of ASEs per local genus, broken down by whether this is a generalist, a migrant, or a resident. Right, so here a migrant, let me back up for a second. A migrant is one of these genera that, let's say, it has a preference for its animal genus, but it has a preference for animals, but sometimes we'll observe it in water. Okay, it's not really supposed to be there. How does it diversify as a function of the water community when it's found? Okay, so that's how we define migrants. So here's what the relationship looks like. In the animal environment, strong DBD relationship for residents, right? So resident diversity is favored, the diversification of residents is favored by higher resident community diversity, with not much of a relationship for generalists or migrants. And similar trends in the other environments, so we always see strong DBD in residents, which I alternatively call habitat specialists. Generalists are pretty flat. And then we actually see a negative relationship for migrants, which I intuitively maybe makes sense, where you have this really maladapted migrant that's coming from animal to water, it arrives there, it's not going to diversify, and there's not much room for diversity to accumulate by migration because it's not happening. So strong DBD residents and some evidence for ecological controls or sort of niche building in migrants. Last question that we thought about is related to genome size. So how is DBD in a lineage related to genome size of that lineage, right? So do lineages with larger or smaller genomes tend to diversify more as a function of community diversity? Again, there's a couple of competing hypotheses from the sort of microbial ecology literature. So one hypothesis is more genes, more DBD. So here, the logic here is that lineages with more genes are better able to survive, thrive, and accumulate diversity in a new community because they can do all sorts of metabolic interactions and cross-feedings and things like that. And there's some literature on this, so we know that in soil, bacteria with larger genomes are able to occupy a wider range of niches. So they kind of have this metabolic flexibility. Maybe there's more biotic interactions that are available because of this. The alternative is something called the Black Queen hypothesis, which was put forward by Morris and Zinser in 2012. Do any of you know the card game Black Queen? Or no, sorry, I'm not a card player. That's not the name of the game. There's some card game, and the goal of the game is not to take the Black Queen. Hearts? Hearts, thank you. Okay, great. So this hypothesis is basically saying that bacteria with smaller and more streamlined genomes are more dependent on others. And so what the black queen hypothesis is positing is that in the community there are some essential functions that are encoded by genes. And as long as the community encodes that function, not every species in the community has to have that function. So it's kind of like this black queen where it's advantageous for every species to basically be a cheater and delete that gene because you can just get the biosynthetic gene product from someone else in the community. As long as the community as a whole does not lose that gene, everyone's okay. But at the individual species level, the selection is to get rid of that product. Okay? So here we might expect that species that have lost more genes are then dependent on the biosynthetic capability of other community members, and they can only diversify in a diverse. Okay? That's kind of the black hole. So this would break this kind of relationship. Okay. So to test this, again, we can just add genome size to our model. We do this at the genus level because that's about where we can get good genome size estimates from GenBank. So we do this, we have a range of genome sizes, pretty much representative of bacteria, so from 1 to 14 million base pairs, and a mean around 5. And we plot this relationship, and lo and behold, we see positive relationship. So this is sort of at odds with the black queen, where larger genomes experience more DNA. And this is consistent with larger genomes with more genetic or metabolic interactions. And I'm trying to find a name for this model, and I just came up with this last night, so I'm running it by you for the first time. It's jack of all trades, where you have sort of more jack of like different suits of jack, right? So it's like you've got more genes that I don't know. They're probably different, but anyway, just trying to. So the opposite of the black queen tradition. Okay. So to summarize this work, so we see that TBD is the dominant model among extant bacterial taxa at nearly any phylogenetic depth we can look at. And so the fact that these effects seem to be strong even down at the phylum level suggests that these deep taxonomic ranks do have some kind of ecological meaning, which at least in bacteria is sort of debated, like what does proteobacteria mean? You know, this is sort of like an ancient group, but do they share useful traits in common? Do they have a real meaning other than just taxonomy? So that struck me as kind of interesting. This is survey data, right? So it's hard to disentangle causes and effects. We can look at possible abiotic drivers. It's really hard to know, and I do see this work as really complementary to the experimental evolution work, where you can really control. You can have a synthetic chemistry, put a bacteria in and see how it diversifies. Really nice controlled experiments. This is the opposite of that. It's a broad survey, so we see general trends. Hard to get real mechanism out of it, but I see this as complementary. We see that DBD seems to reach a plateau as niches are filled. And as these niches fill, abiotic drivers of diversity do take over and become more important. And in terms of mechanisms, as I said, we're a little agnostic to mechanism. In the genome size analyses, we get some hints that genetic capabilities, crossfeeding, niche construction could be important. Okay, so I have a bit more time, and I'm really excited about this because all this is in progress, and this is very much in progress. So I'm taking this opportunity to sort of float some ideas here. So so far, as I said, we're kind of considering mainly a community assembly model at the resolution of this marker, right? Or a little bit agnostic. A bit hand-wavy about what I mean by diversification, and this is really more of a community model of what's going on. So can we really zoom in and talk about evolutionary diversification in real time, really in the sense of actual accumulation of new mutations, polymorphism within species. Okay, so how to do this? let's look in the human gut where we have a few nice times here. So this is using data from Eric Hall from my old lab, a paper that came out recently, where we've got one person sampled in poop samples of human gut microbiome, stool samples monthly over 18 months. And we're going to look at diversity within the species Bacteroides vulgaris, which just happened, is just convenient because it's quite abundant. So using shotgun metagenomic data, right? So just sequencing all the DNA in the gut, we can have a lot of sequencing reads that align to this genome, get a good depth of coverage, and be able to call single nucleotide variants. So that's what we did. I won't get into all the methods, but we can track simultaneously from metagenomes the overall community diversity, so the species richness in the gut over time, about 18 months. Compute the Shannon diversity. And then in our focal species, you can count the number of single nucleotide variants, of SNPs. And here we use the very wise filters from Nandita's paper from a couple of years ago. Still working on how different filters, how this affects things, but you can see that the range is not very high, right? So we see between 0 and just 17 or something like that, a point mutation. So this is like a point mutation within a species. And I should say, right, so this is over time, right? So this is always, in this case, this is like the number of single nucleotide variants that have appeared relative to the time point before. Okay? So then to look at our DVD pattern, we basically are going to want to plot this diversification against the community pattern. And already you can see there's a lot of sort of spikes here. And already you can see that sort of the rolling average here, this is like a low S, smooth. There's a little bump here, and it kind of tracks in phenol gas as well. And disclaimer, n equals 1, one person, one bacterial species, right? So it's all really exploratory. But if you plot diversity, community diversity against diversification, we do see positive slopes. So not huge correlation coefficients, but the tendency does seem to be positive. So there does seem to be a temporal signal of TDE at the level of point mutations, so single nucleotide variants within this one bacterial species in this one healthy person. Okay? So that's interesting. The other cool thing that we can start to do with metagenomics, right, is genome-wide, it's not just the marker gene, we can look at gene being lost. So again, using a coverage filter in a reference genome, is the gene present or absent in that particular time point. So just looking at loss events, so again, this is like from at time 1 relative to time 0, counting the number of gene loss events. And here there's no relationship between gene loss and either richness or Shannon diversity. Also note that these scales, and we can talk about this later, and if you're interested, but these are a lot of genes for a bacterial genome, right? So this is a genome of like 5,000 genes. So these are a lot of genes. So these are, from my perspective, probably not genes actually being lost. These are just different strains of B. vulgaris that diverged a long time ago that have different gene contents, unless it's all like a plasmid or something. But okay, so no pattern of gene loss. But if you look at gene gain, there's actually like a really strong pattern from my perspective. Reasonably strong, which I think is pretty interesting. So my interpretation of this is that community diversity select for or favors B. vulgaris strains. Sorry, community diversity causes B. vulgaris strains with fewer genes to be replaced by strains with more genes. This again kind of resonates with genome size analysis I showed before. It's basically like having more genes or more diverse communities will favor strains with bigger genomes and more genes. And then at the finer level, community diversity will cause B. vulgaris to diversify by point mutations in terms of SNPs. So, as I said before, the scales of these SNPs, this is a reasonable number of point mutations to be happening de novo. This, I think, is sort of at the level of strain replacement. So I think we have both patterns happening simultaneously. Okay, so that's kind of my like hot off the press, fresh result. This is kind of the direction we're going. You know, we'd like to expand this a little more, but you know, each of these analyses, even looking at one species in one person, it's like quite a lot of data to crunch. So starting modest, and we'll build up from here. That's all I wanted to tell you today. So I want to thank very much PhD student, fantastic PhD student, my lab, Naima Mahdi, our collaborator, Mick in Exeter, Pierre Lejean in my department, who's written textbooks on stats for ecology, and funding from the Natural Sciences and Engineering Research Council of Canada, Canada Research Chair. This really nice to have this kind of funding to do very basic and exploratory kind of work, so I'm very grateful for this. And other members of my lab who contributed through discussions and are just a great group. This is us at one of our retreats on a frozen lake, so we don't get much of that here, but we enjoy the winter. And this is the same lake in the summer, so you know there is summer. And I will close by with a pitch that if any of you are undergrads or master's students looking for a PhD or looking for postdocs, my lab is recruiting. LA is pretty great, so I feel like this is a hard sell. Coming from Montreal in February, I won't show. This is like Montreal in autumn, where this is the Mount Royal with lovely trees and things like that. But it's a great city for students. I'll include postdocs as students as well. Cost of living is very reasonable. It's a really fun city, and you're at one of the first groups that I'm announcing. That my lab is moving, still in Montreal, so don't worry if you want to come. We're still just on the other side of the mountain here at McGill, from the University of Montreal to McGill. I'll be joining the McGill Genome Center and the MI4 initiative in infection and immunity. So shoot me an email if you're interested in opportunities, and happy to take some questions. Am I choosing my own questions? Okay, let's start there.
I'm just curious, thank you for the talk. I'm just curious to know if those gains are duplications or those novel functions. Do you know? Have you explored that already? Or?
I think that they are novel genes in the sense that we're not considering like copy number variation on the same gene. This all comes down to the way that you do the filtering for coverage. I think that the way we've done it is that it's not.
What is the nature of competition in these diverse communities that might limit the diversification of your local groups? Is it like exploitative competition where resources are being utilized more efficiently, or is it interference, whether they're poisoning each other by antibiotics? It seems all complicated because they're exchanging genes too, right? I mean, so I really don't understand if you were to drill down at a very mechanistic level what might be going on.
That's right. And I think that in some of these experimental evolution studies, they have been able to drill down at mechanism. And this is like a 1,000-foot view. So I think all those things are happening to some extent. There's some interesting experimental work showing that competition can actually lead to increasing diversification because it causes underutilized resources to be used, so that you get niches that otherwise are ignored that start to be filled because of this competition. So yeah, but certainly in terms of interference competition, killing by antibiotics, type 6 secretion systems where bacteria are stabbing each other, and all this stuff's going on, but I don't have too much intuition of which of those are. Yes?
Yeah, I'm curious what kind of differences you saw between the foregut and the hindgut samples. Because I know it's hypothesized that there's more close controls in the foregut.
Right.
Because that's where the most nutrients are. So I'm curious if you saw differences between the two.
Interesting. Yeah. So there's all these. Yeah. So I have it. I'll look at that. Or feel free to go in the preprint and look. There's all this. We have like a lot of supplementary graphs that show all of that. That's cool. I'll have it.
Yeah, I'm trying to understand what diversification is in bacteria. And so, can you explain to me how your results are not reflecting that more habitats that have more ecological opportunity are just not filled by more species, and fewer species are able to diversify into habitats with fewer niches?
Right. So is this just that some?
We can have more bacteria in some places than others.
Oh, in terms of like actual biomass.
Yeah. No, not the number, not the individual numbers or whatever, but like species. Oh man. I am trying to understand if you're really tracking diversity begets diversity, or if you're just seeing that you can have in some habitats you can just have species and others.
Right. Which is, I think, to some extent, this circularity between like some environments, some samples are just more diverse. But I guess the subtlety is that it's that is not, I think, not trivial. Is that okay? Yes, and we know some like soil is more diverse, right? For example, right? So some samples are more diverse than others. But what we're seeing is, and it's hard to get at mechanism, but there's a scaling, being very agnostic towards is this diversity begets diversity, but just the observation that there's a scaling of diversity at different levels. So sort of at like the level of genera in the community and the number of species per genus.
Because in a
which is not, I think, not
in a multi-cellular macroevolutionary context, there's these ideas often include an important component of time. Like you imagine that there was an empty set of niches and that they are filling over time.
right.
And it's not clear to me, well, it seems like your sampling is well past any phase of filling. Aren't these are supposed to be the sort of equilibrium?
Yeah, right. And this is where I think it comes back to at the resolution of 16S genes, like these are ASVs or species that have diversified over the past many millions of years.
Yeah.
And now we're saying, how are they distributed across extant samples, right? So it's, and I think there are some interesting things where, and this is maybe a bit hand-wavy, but that it seems that in animal, so host-associated, animal-associated biomes are still on this sort of upward trajectory where they're not very diverse, and there is seemingly a DVD, like a positive feedback, whereas soil and water, which are older, predating the evolution, and this is like deep evolution or predating the evolution of animals, are very diverse and kind of just at this equilibrium. But maybe animals, and this is really on like fairly deep evolutionary timescales, are younger.
Yeah.
And so they are on a different part of that curve.
So are the things that you're the differences between genera that you're seeing, are they on the scale of the diversification that you see in the experimental bacterial evolution papers, or are those experimental bacterial evolution differences between whatever species much smaller? Like what's this?
Yeah, no, much smaller. So this is where like this is more like the scale of the experimental evolution, where you're seeing like a dozen point mutations versus what we're tracking with these 6S amplicons are tracking like hundreds or thousands or tens of thousands of point mutations. So really, really deep.
So do you have an idea what the diversification rate is for any of these taxonomic levels?
At the level of 16S, it like on average, and of course it will vary across the tree to some extent, but it is like the clock is something like 1 point mutation per million years in 16S or something.
Oh, sorry. We can talk more.
Okay, yeah.
Thank you. Yeah.
I have one quick question. It's great to hear. Thanks so much. I was wondering about maybe a little bit related to what Mike is talking about. We go to the first part of the talk. How much of that can be explained by just extinction rates? I know that the extinction rates are difficult to estimate
Right.
but
Yeah.
how much extinction rate are really?
Yeah. I mean, that's fascinating for bacteria because, right, so all of this is the balance between speciation and extinction. And again, the reason that we didn't, at least so far, go for a whole phylogenetic method, just going for taxonomy, and then I guess a pseudo-phylogenetic method of just using a percent identity similarity in 16S sequences, is that we don't have fossils for bacteria, so the phylogeny is not well calibrated. We can't track this. We don't have fossil records that something was here and then it's extinct. So my feeling, and maybe I can be corrected on this, is that more detailed, explicit phylogenetic methods are just going to be, the inference problem is going to be too hard for bacteria, at least for bacteria at the level of the tree of all the bacteria. Like you could do it in this kind of context where you just have like one species and you have a few mutations you can place on a tree. That's fine, but for the entire tree of life of bacterial diversity, it's just too hard to infer as well. But maybe I'm wrong.
Yeah. So I'm from the environmental engineering, so I learned some paper in the soil environment that maybe the higher diversity will contribute to more about ecology, stability. But in your study, you found that the ecology control will reduce the diversity. Do you think is there any conflict or there's balance between these two terms?
Yeah, no, exactly. So I think I set up like the opposing models, but I think that, so my sort of mental model is that initially there is a positive feedback of diversity, you got diversity, and then eventually niches get full. You can't have a positive feedback forever. This kind of explosion of diversity forever. Eventually there for sure will be a finite number of niches, and then it just plateau. That's sort of my my interpretation.
Okay, thank you.